Copyright Stanford University, all rights reserved.
Far here from UC Irvine, who will be participating later in the program, with the idea that we can try to use this as a forum to make sure that we're letting everyone know about myotonic dystrophy, where it is in terms of research, where we're going in terms of clinical care understanding, and also to get more involvement of the community. So it's great to have you here as we launch what we hope is going to be an ongoing session. Stanford dictates that I share with you some of my conflicts of interest, so you can dismiss anything you think is biased in my presentation. I don't anticipate this being a conflict, but now you know what they are. So basically what I'm going to do, and I was playing with the pen here earlier, so I've left marks that maybe I can erase. I know, isn't that cool? I was having so much fun with it. So I mean, the good news, many of you have heard me talk before at various meetings or in clinic or whatever. So the good news is you're not going to have to hear me drone on much. We've actually got some bona fide experts here this time, so you don't have to hear me try to pretend I know anything about RNA processing or gastrointestinal function or something. That we actually have some people here who know about all this stuff. And so I get to learn along with you. So I'm just going to introduce kind of the concept, and then we'll invite up Dr. Aries from UC Santa Cruz, who is a worldwide recognized expert in the type of RNA processing problems that go on in myotonic dystrophy. And if that doesn't mean anything to you, hopefully it will by the end of his talk. So we do have a number of exciting people to hear from today, which will, I hope, be of value. I've chosen a few of the topics of clinical issues. Oh, there I did. I was playing here too, I guess. To talk about in terms of clinical care. And I just selected 3 out of the air. There are many, many, many issues clinically in myotonic dystrophy. And so I just picked 3, and we'll end up talking about those. But I did want to undergird that with a discussion a little bit about the genetic mechanism, and talk a little bit about where we're heading in terms of what we hope will be a meaningful clinical trial that at a very, very limited level we should be launching this year. We've got a lot of work to do before that trial is going to be effective, and that's where we need to partner with the entire myotonic dystrophy community, which is another reason that we're excited to see you here today, because this is not something that can just be done in the lab or done in the clinic without the involvement of the myotonic dystrophy community. So we really do need to be able to connect with you. So the more you can get involved, the better, and we'll talk a little bit about that. So just to kind of give a little foretaste of what underlies myotonic dystrophy, we'll just go through this a little bit. Many of you know your repeat number. Oftentimes, if you're affected, you know your repeat number. And what that is, of course, is in your DNA, in this one gene, that there's an increase in either CTG, if you have myotonic dystrophy type 1, or CCTG, if you have myotonic dystrophy type 2. And in essence, the problem that causes is that you may remember from 10th grade biology that DNA is turned into RNA or reread as RNA, and that RNA folds back on itself and creates these pink dots. Now, they're not really pink. I mean, they're labeled here. They're colored pink, but they really are in every one of your nuclei, so that they're present in all the nuclei of all the cells in your body, basically. And those end up being an interesting phenomenon. They were actually identified long before anybody knew what they did. So here, back in 1995, shortly after the gene was found, the people working in this MIT lab found these clumps of this RNA. So the RNA was binding up and clumping in the nuclei, but it wasn't clear whether that was just happenstance and a phenomenon that didn't mean anything, or if it was really related to the disease process. It turned out then, when we found myotonic dystrophy type 2, that we got these same clumps. And the fact that those clumps were present in both diseases made all of us think that maybe they really were involved in the disease process. And that was further evidenced by work, in this case out of Charles Thornton's lab, working with Maury Swanson, identifying a protein that bound to the RNA. So that we don't need to get too into this, because fortunately we have somebody here who actually understands this stuff who's going to talk next. But the bottom line is that having this long repeat of CTGs ends up causing those pink clumps, if you will, to which you get this protein stuck, and then the protein's not out there doing its regular job. So a simple-minded approach to treating this would be that if we can get rid of the clumps, we could get rid of the disease. And so that is where we're at in terms of trying to develop a treatment for myotonic dystrophy. And this is one approach that we'll be taking in this trial as part of a myotonic dystrophy research network that involves the University of Rochester, Ohio State, University of Kansas, and University of Florida, as well as Stanford, for the initial trials. And then as those get bigger, I'm confident that we'll need to involve more centers like UC Irvine and other places that are also interested in pushing these new treatments forward. So that the bottom line here is that if you have a different type of RNA that's kind of coded differently, it sticks to those clumps and it can help eliminate them. And so it's simple-minded, but it looks like it actually might be effective. It certainly seems to work well in the mice, and there's some evidence that we can do this now in people, because we're doing similar kinds of trials in people, and it looks like this little tiny synthetic type of nucleic acid gets into the cells and does what it's programmed to do. So we're optimistic about it, but we don't think that this is likely to be a cure. We think this is, if we're thinking right, that this is going to move us in the right direction towards slowing down the disease process, eliminating some of the features, and then we'll come back to it and try to refine that and amplify that. So this is a marathon. This isn't just a 100-yard sprint here, that we're going to have this all done and locked away within the next year or two. This has got to be a long-term process, and we're going to keep going. But we are nonetheless quite excited about this possibility to begin treating this disease right at its root cause. And this just shows you the work that Charles has done in his mice, Dr. Thornton's lab. And here again, if you can see it, here are some pink dots in these mice that he programmed to look like myotonic dystrophy. And then when he treats it with this so-called antisense oligonucleotide, and that's, if you don't remember anything, maybe you remember that the letter is ASO, or I don't know how you're going to remember this. There's no like catchy term for this, is there? I mean, I don't know, gene bullet or something. I don't know. It would be nice if we came up with something that was an easier handle for this. But nonetheless, you can see that the pink dots are gone over here. And so that's the good news. And it's pretty, but it ends up that it doesn't just have an aesthetic kind of treatment. It's not just cleaning up the pink dots, because when he looked at his mice, it turns out that one of the proteins that was secondarily absent because of those pink dots came back. So these red, maybe this is too bright. That's part of the problem. Does that do anything? Did it do something? How about this? Whoa. All right. It doesn't like me to do that, huh? How did that happen? All right. And now this gets into the clinical features of myotonic dystrophy. If half the audience falls asleep, then you know, then we're in trouble. So I will keep flashing the lights back on. But you can see here, this is the programmed mouse that has myotonic dystrophy. And then when he treats it, the red protein, in this case, he's looking at a protein that causes a chloride channel in muscle, comes back, and it looks almost like the normal mouse over here. And what that ends up doing then is getting rid of the myotonia. So this is just a measure of myotonia, that tendency for the muscle to stick or not relax, and it's basically gone in the mice after they are treated. And so there's reason to be optimistic that we're going to be able to control this. I'll wake you back up. If this works, and that's just a marker. It's not like this is only to treat myotonia. We hope that it actually has many other effects on the muscle and on other organ systems beyond muscle, but we're not going to really know what it does until we get in there and start studying it. So this is just a cartoon showing one version of how this works at a simplistic level that Dr. Aries is going to be able to tell us more about. But normally that protein, it's called muscle blind, that gets stuck in those pink clumps, is responsible for helping process the RNA from a number of different genes that end up being important for heart function or metabolic function or muscle function, and that with myotonic dystrophy, the clumps being present gets rid of that protein, so it's no longer available to bind to these genes, and consequently ends up causing an abnormal protein and various specific features of myotonic dystrophy. So that's all I was going to say about the biology of this. And I know it's really going over a lot very quickly, but we'll have time to talk with Dr. Aries and others and answer some questions here in a little bit. So we know that there are many features in myotonic dystrophy, and we have some ideas of some of the genes that are altered, but Dr. Aries' lab and others are really helping us understand just how many changes there are in these processing functions. And then we can attack the symptoms of myotonic dystrophy directly, either through that antisense oligonucleotide or through altering specific genes. All right, so again, I've given myself 5 minutes to go through the clinical features of myotonic dystrophy. So obviously, this is going to be rather truncated as well. But as you all know, myotonic dystrophy is a whole lot more than just a muscle disease. It affects basically every organ system. It does a lot of different things. And what you don't want is, if you have a doctor that you're following up with on an annual basis for your myotonic dystrophy, is to just go in and have them say, yep, you still have myotonic dystrophy, because that you could be pretty assured of. It's very unlikely that it'll just go away in an annual visit. And so you really have to go beyond that. You really have to have someone who understands the complexity of the disease, and that's why going to a physician like Dr. Mozaffar or the members of our team here or any of the other teams more familiar with myotonic dystrophy ends up being so important in terms of controlling all the features that are part of this disorder. So this just shows some of the patients that I've worked with. They can be very severely affected or moderately affected or mildly affected, but in almost all of them, all of these tissues or organ systems are affected if we look at it. If we look more closely, we can see some changes, sometimes subtly, sometimes very dramatically. not only are they affected, but they're affected in different ways, so that you can have early onset problems that don't change that much, or we can have problems that come on later in life and then get worse as time goes on, or you can have problems that really just kind of fluctuate through life, or we can have a whole lot of different elements of the disease, all of which are part of this very, very, very complex disorder. And then each of these different processes can affect each of the different tissues. So we can have muscle features that develop early on and then stay relatively static. We can have muscle features that come on later in life and then progress. We can have muscle features that just fluctuate throughout life. We can have a lot of different effects on muscle and on brain and on heart and on eye. And you can see how this gets really, really complicated. So that's why it's really helpful to us to have a group of physicians involved. And it's exciting to me that we're going to have some experts here later today who can talk with you about the heart effects and the sleeping effects and the gastrointestinal effects, to just take 3. All right, so now I wanted to ask you some questions so that we can I can learn something from this as well. So for those of you who either have myotonic dystrophy or have myotonic dystrophy in your family, if you can think, so if you yourself don't have it, think about somebody in your family and see if you can answer questions with them in mind. Okay, so the first one is with regard to gastrointestinal function. So everybody can vote on this, so that you don't have to have myotonic dystrophy, but you can be thinking of different people in your family. So how many have had recurring stomach or bowel problems? Okay. Okay. You're counted. So a pretty high number. I'd say the vast majority of people have had some stomach or bowel problems with myotonic dystrophy. How many have actually gone to see their doctors and discussed their stomach and bowel problems at some point? So somewhat fewer, but still quite a few. And how many have actually been given a treatment that their doctor recommended for their stomach or bowel problem? Okay, the numbers are going down some, but now we're getting down to a handful. And how many responded well to that treatment? We've got 2. Okay, so we need to find out what you're on, but the most important point in that, and we'll pass that on to Dr. Wen, who will be talking with us about gastrointestinal function, is that we've got a long way to go to understand what's going on with the bowel. The bowel is clearly affected in myotonic dystrophy, and we clearly don't really understand it all that well. We have some ideas, but we need to do a lot to get a better treatment. So let me just move on to the heart. So Dr. Perez is going to talk with us about one aspect of heart function. And so again, you can answer for yourself or for any family member, but for people with myotonic dystrophy, how many have ever had an electrocardiogram? So pretty much everybody, I think. Good. And how many have had an electrocardiogram done pretty much every year? Good. So we're doing pretty well. That's certainly a recommendation, because we want to be able to monitor not just whether or not the electrocardiogram is normal, but even if it's changing within the normal range. That's helpful for us to know. Certainly, if it gets abnormal, Dr. Perez is going to want to know about that. How many have been told that they had some type of abnormality in their EKG or ECG? And again, it's not uncommon. So certainly in myotonic dystrophy, it is not at all uncommon that we'll see some abnormality. We don't always know what it means in terms of function, but it's often showing up. How many have had a Holter monitor, if you know what that is? Excellent. So that ends up being another important way of investigating. A Holter monitor is something that you wear for a couple of days, and it records your ECG signal so that Dr. Perez and others can look at it and determine whether or not there's any recurring abnormality. Maybe it's not there all the time, but it pops up every now and again during 48 hours. How many have been put on a treatment for heart irregularities? Now the number is going way down. And how many have had a pacemaker or a defibrillator placed? 1, 2, 3. So now the number has gone way down. And that's partly because, I mean, and Dr. Perez can talk about this, but, you know, I think we're still exploring, still learning a lot more about when that ends up being important. I'm certainly not saying everybody needs a pacemaker and a defibrillator. But I think that, you know, we need to work with you. And again, this is part of the goal, is that if we can work with you, we can define these things better so that if we can all work together, we can get better standards and know how to deal with this. Okay, and then the last issue we're going to talk about is sleep. So again, everybody can answer questions if you have myotonic dystrophy in your family. How many have had or have been told they have increased sleepiness? It's pretty uniform. Okay, how many at some time have been told that they should use some kind of breathing support at night, CPAP or BiPAP? Again, pretty high numbers. How many actually tried the CPAP or BiPAP? Okay, so we lost a few. How many are continuing to use their CPAP or BiPAP? We've lost almost everybody. We're down to like 3 or 4 that have tolerated it. And I think that is certainly my experience, that it has been really, really hard to stay on that if you need it. Okay, how many have been prescribed a stimulant medication to help with daytime sleepiness? So that's like Provigil or Nuvigil or Ritalin or Cylert. Pretty high numbers. How many tried it? Pretty much the same number. And how many have continued to use it? Pretty much the same number. So you'd be interested to know that the clinical studies on the effects of stimulant medication on daytime sleepiness show it doesn't work, which to me shows that the studies were wrong. I mean, you know, because the overwhelming experience has been that it does. And, you know, I think that's a good indication of the kinds of problems that we have. We can do these studies, but if we don't know the right thing to measure, we're not going to pick up the effects. And that comes back full circle now to the genetic treatment that we're talking about doing. We have to go into that study with a really good, really strong marker of the benefits that we think we're going to see with that genetic treatment. Otherwise, it'll look like it doesn't work. And the FDA, we can sit there and say till we're blue in the face that we think that this helped our patients, but if it doesn't show up on the marker we choose, and we have to basically choose one marker, then the FDA is going to say it doesn't work. You told us it would affect this, and it didn't. And so if we have the same experience with the genetic treatments that we did with the stimulant medication for sleepiness, we're in trouble. And so that's why we really need you to work with us to improve the outcome measures and get testing so that when these genetic trials start, we're actually testing something that we think is going to be a bona fide marker of what we're treating. So we can only conquer this disease if we're working together, which is why I'm really thrilled to see all of you today, and I look forward to kind of pushing this forward in every year to try to help the myotonic dystrophy community help us so that we can help you. And so it's very much got to be a team effort. There are a lot of things that can be done, and we'll be having you sign up or give you the opportunity to sign up for a number of these things. I encourage you to go on the website for the Myotonic Dystrophy Foundation and join the registry so that you can get on their mailing list That's not the same thing as joining the registry. Joining the registry, there's a questionnaire to fill out. It's quite simple. It's very well organized on the web, and it gives us some insights into myotonic dystrophy while also increasing our ability to contact the community. There's a different registry, just to keep this clear, at the University of Rochester. It's a different type of registry. It's for a different purpose. They're both valuable, and I strongly encourage you to participate in both if you can. You can contact the National Registry at the University of Rochester, or you can get it from our clinic when you come in. We can help you find that. I think it's helpful if the myotonic community goes and sees people who are aware of myotonic dystrophy and can get you involved in research and get your care optimized. So there are a number of centers around the state that do that, and I think it's great. The more we can, our centers are getting very closely networked so that when Dr. Muzaffar is doing a study, we're helping him. When we're doing a study, he's helping us. And that allows you, so you don't have to all go to one place. We can establish a network so that we can all interact and get these studies done and move the ball forward. So there's also a questionnaire so you can help us learn how to make this meeting better next year. And then there's another list that you can check off if you're interested in any of the research studies that we have. And here's a list of research studies that we currently have ongoing at Stanford to investigate